Hi, this is Alex from devstories.eu. Biopython Fundamentals, Episode 15 of 18. When you submit a paper describing a newly sequenced bacterial plasmid, reviewers rarely want to read a plain text table of gene coordinates. They expect a cleanly annotated, colored, circular map that makes the genomic structure obvious at a single glance. Visualizing genomes with GenomeDiagram is how you generate those maps directly from your sequence data. Before you start drawing, you must ensure the external Report Lab library is installed. GenomeDiagram relies entirely on Report Lab under the hood to calculate the geometry and render the final images. Without it, the graphical export will fail. The architecture of GenomeDiagram is strictly hierarchical. You construct your visualization by nesting objects from the top down. At the absolute top is the diagram object. This is your blank canvas. Inside the diagram, you add one or more tracks. A track represents a specific plotting area. On a linear map, a track is a horizontal row. On a circular plasmid map, a track is a single concentric ring. Inside a track, you add feature sets. A feature set is a logical grouping of sequence data. You might use one feature set for coding sequences and a different feature set for regulatory regions on the exact same ring. Finally, inside the feature sets, you place the individual sequence features themselves. Here is the key insight. You do not have to manually calculate angles or pixel coordinates to place your genes. GenomeDiagram reads the start position, stop position, and strand orientation natively from standard Biopython feature objects. To put this into practice, suppose you have a parsed GenBank record for a small plasmid. First, you initialize an empty diagram. Then you create a new track inside it and a new feature set inside that track. Next, you iterate over the features in your parsed GenBank record. You check if the current feature type is a CDS, meaning a coding sequence. When you find a CDS, you pass it to your feature set to add it to the visualization. During this step, you assign visual styling. You can tell the feature to render as an arrow, which automatically points clockwise or counterclockwise based on the gene strand. You can also assign distinct colors here. A common trick is to use a simple loop counter to alternate between two colors so that adjacent genes do not visually blend into one massive block. Once your hierarchy is fully populated with data, you instruct the diagram to draw itself. You call the draw method and set the format to circular. You can define the page dimensions, the background color, and the track scaling. After the drawing is constructed in memory, you simply call the write method, passing a file name ending in either PDF or PNG. Report Lab instantly calculates the correct arc lengths, positions the colored arrows along the circular track, and exports a publication-ready file. The primary advantage of this tiered structure is reproducibility. Because the layout rules are strictly separated from your raw sequence parsing, you can write one script to uniformly style an entire database of plasmids without ever touching a graphic design tool. Thanks for tuning in. Until next time.